stem in dicot and monocot. So in dicot, the stem is covered by an external covering which is present above the epidermis, which is thick and waxy, and it is called as cuticle. In open vascular bundle, between the phloem and the xylem, there is the cambium that is present, and these cambium that is there will help in the formation of the secondary tissues, that is secondary xylem, secondary phloem and all that. Now you have to differentiate between the two with respect to ground tissue system. Ground tissue system means with respect to the cortex we have to differentiate. Hello everyone, a warm welcome to this session on revision class for first PUC Biology. I am Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So in this session, let us do revision on the chapter that is Anatomy of Flowering Plants, wherein we'll go with the following contents, that is synopsis. So what and all is there inside this chapter, we'll have a look into, and then we'll move on to learning some of the important MCQ, that is multiple choice questions, and 3-mark question and answer. So from this chapter, one multiple choice question will be asked and one 3-mark question will be asked. Therefore, it carries a total weightage of 4 marks. So starting with the synopsis, so we have to learn about the tissue system. So tissues are nothing but a group of cells. So what are the different tissue systems that are present in the plant? Starting from the beginning, that is the outermost tissue layer covering, it is the epidermal tissue. And in the epidermal tissue it consists of the cuticle, the epidermis and the stomatal apparatus. So cuticle is nothing but a waxy coating which is thick and it is present above the epidermal cell layer or epidermal layer. And in the epidermal layer, some of the cells of the epidermis get modified to form bean-shaped structures called as stomata in the case of dicots, and if it is monocot, it will be dumbbell-shaped structures. And the stomata are very important for transpiration, for respiration and all that. Next, below the epidermal tissue is present the ground tissue, which are made up of few layers of cells. For example, it is called mesophyll. It is made up of mesophyll cells. In this mesophyll cell, it consists of parenchyma, collenchyma and sclerenchyma. So parenchyma are those oval-shaped cells which are loosely arranged. Now those oval-shaped parenchyma cells, if are tightly packed because of some depositions, then we call it as collenchyma. And those parenchyma cells which has chloroplast in them, we can call it as the chlorenchyma. And sclerenchyma, these are fibrous tissues which actually provide mechanical support to the plant. So these are the cells that come together to form the ground tissue system in the plant. Next, below the ground tissue system or the innermost layer of the plant is nothing but the vascular tissue. In the vascular tissue, it mainly consists of the xylem and the phloem, and depending on how they are arranged, it is of 2 types, radial and conjoint. Radial and conjoint. So conjoint, it can be open conjoint and it can be closed conjoint. So conjoint is of 2 types, that is open conjoint and closed conjoint. Next, moving further, talking about the anatomy, that is how exactly the cells are arranged or the tissues are arranged inside the plant. So first we'll talk from the below level, that is root, then we'll move on to the aerial level, that is the stem, and then the leaf. Talking about the root, in dicot and monocot, there is a difference in the arrangement of the tissues in the root. So in dicot, they have epiblema. The outermost tissue forms the epiblema, that is the epidermis. And below the epidermis it consists of cortex, and that cortex is made up of entirely parenchyma cells. And below the cortex is present a layer of cell called as the endodermis, and in that endodermis they have some Casparian thickenings which are present in the tangential and radial wall. So Casparian strips will be present. That is one of the characteristic feature of dicot root. Next, pericycle will be present below the endodermis, and talking about the vascular bundle, they have 2 to 4 xylem and phloem patches will be present. Next, talking about the monocot. Monocot, they usually vascular bundles, they are polyarch. Polyarch meaning more than 6 vascular bundles will be, xylem bundles will be present in them. And this is one of the characteristic feature of the monocots, and they have a very large pith when compared to the dicot. So next, talking about the stem. So stem in dicot and monocot. So in dicot, the stem is covered by an external covering which is present above the epidermis, which is thick and waxy, and it is called as cuticle. Below the cuticle is present the epidermis. Below the epidermis is present the cortex, and in the cortex it is made up of collenchyma cells only, wherein the parenchyma cells, which are closely arranged with depositions, we call it as collenchyma, and that layer which consists of the collenchyma cells, that cortex layer, is called as the hypodermis. Next comes below the hypodermis, there is presence of the endodermis, and this endodermis is also called as the starch sheath in the case of dicot stem, because these endodermis, they have depositions of starch in them. So that is why it is also called as the starch sheath. Below the endodermis is present the pericycle, and the vascular bundles are present wherein xylem and phloem is present, and their arrangement is in ring-like manner, and they are open conjoint vascular bundle. So open conjoint vascular bundle means between the xylem and the phloem there is a cambium ring that is present. So that is open conjoint. Closed means there will not be cambium ring that is present, and they have a very large pith. So these are the characteristic of the dicot stem. Now moving to monocot stem, they have the cortex, and below the cortex here also we call it as hypodermis, but the hypodermis is made up of sclerenchyma cells, whereas the hypodermis of the stem, dicot stem, is made up of collenchyma cells. The hypodermis of monocot stem is made up of sclerenchyma cells, and the vascular bundles are present, but they do not have a radial arrangement of vascular bundle, while the vascular bundles here are quite scattered, wherein the larger ones are towards the center and the smaller ones are towards the periphery. So towards the pith, the vascular bundles are larger. Next, the type of vascular bundle that they have is conjoint closed. So conjoint closed vascular bundle means there is, so conjoint closed vascular bundle means there is no cambium ring between the xylem and the phloem. So this is about the dicot and the monocot stem. Next, moving to dicot and monocot leaves. So let's look into them. That is dicot and monocot leaf. So dicot leaf, they have cuticle, and below the cuticle is present the epidermis. Then they have the layer of parenchymatous cells, and that region is called as the mesophyll. In the mesophyll, they have parenchyma cells, wherein they have spongy parenchyma as well as palisade parenchyma that is being present, and the vascular bundles are surrounded by a bundle sheath. These are the characteristics of the dicot leaf. Next, moving to monocot leaf, they have the epidermis and they have bulliform cells. So some of the epidermal cells, they get modified into usually those epidermal cells which are present near the midrib. So they get modified into large colorless cells called as bulliform cells, and the main function of the bulliform cells is to prevent excess loss of water during summers and all that, so that the leaves will just curl up and it will reduce the surface area of the leaf, thus preventing excess loss of water. So this is the function of the bulliform cells, and they are found only in monocot leaf, especially in grasses. Next, let's look into the multiple choice questions. What is the function of the cuticle? So cuticle, where it is present? Above the epidermis. So the main function of the cuticle is it absorb water and minerals, regulate gaseous exchange, prevent loss of water, none of the above. The main function of the cuticle is to prevent loss of water, because thicker the cuticle, then the transpiration rates are lower. So next question, where are stomata present? Roots, leaves, stem, all of the above. So stomata usually are present in the epidermis of the leaves, wherein in dicot leaves the stomata are usually found in the adaxial epidermis, that is the lower epidermis, and in monocot they are found equally distributed on the adaxial as well as the abaxial epidermis. So in dicots, abaxial epidermis is the region where the that is lower epidermis is the region where more number of stomata will be present. So that also question can be asked. Next question, what are the two bean-shaped cells that enclose the stomatal pore called? Are they called parenchyma cells, guard cells, subsidiary cells, trichomes? So we can eliminate parenchyma cells and trichomes. Now there is a confusion between guard cells and subsidiary cells. Subsidiary cells are those epidermal cells which surrounds the guard, that is the guard cell. So therefore, the two bean-shaped cells which have a stomatal pore in between them, we call it as the guard cells. So they are bean-shaped like this. So we call it as guard cell, and this is the stomatal pore that is present. So therefore, the answer is guard cell. Moving further, what are the specialized cells in the vicinity of the guard cells called? So what are they called? I told you earlier, subsidiary cells. So vicinity means close to. So those cells which are present close to surrounding the guard cells, we call it as the subsidiary cells. So next question, what are the vascular bundles in dicotyledonous stems called? Is it closed vascular bundle, radial vascular bundle, open vascular bundle, secondary vascular bundle? Usually they have closed vascular bundle. Next question, what is the function of cambium in vascular bundle? To form secondary xylem and phloem tissue, to absorb water and minerals from the soil, to produce chlorophyll for photosynthesis, to transport sugars from the leaves. Its main function is to form secondary xylem and phloem tissue. So that is the function of the cambium. Option A is the right answer here. So next question, which type of vascular bundles do monocotyledonous have? Do they have open vascular bundle, secondary vascular bundle, closed vascular bundle, radial vascular bundle? So monocots have open vascular bundle. So usually monocotyledons, they have open vascular bundle. So next one, what is the location of phloem in conjoint vascular bundle? So only on the outer side of xylem, only on the inner side of xylem, alternating along the same radius, jointly situated along the same radius. It is only on the outer side of the xylem. So below will be present the xylem, then above the xylem is present the phloem that is distributed. So that is the location of the phloem in a conjoint vascular bundle. And alternating along the same radius, it will be a radial vascular bundle. So this option is not correct. It's option A which is correct. Next question. Which type of vascular bundles possess the ability to form secondary tissues? Is it radial, closed vascular bundles, conjoint vascular bundles, open vascular bundle? So which type of vascular bundles possess the ability to form secondary tissues? Is it radial vascular bundle, closed vascular bundles, conjoint vascular bundles, or open vascular bundles? Now here it is not radial vascular bundles. In radial vascular bundles, they do not have cambium. So this option can be eliminated. It's not closed vascular bundle because closed vascular bundle means they do not have cambium. So no cambium means closed vascular bundle. Now the option is between conjoint vascular bundle and open vascular bundle. Of course, in conjoint vascular bundle, you can have cambium or cambium may not be there. So you have to be specific. Here we have to choose the option open vascular bundle because in open vascular bundle between the phloem and the xylem there is the cambium that is present, and these cambium that is there will help in the formation of the secondary tissues, that is secondary xylem, secondary phloem and all that. So therefore right answer for this is option D, open vascular bundle. So next question. What is the outermost layer of the dicotyledonous root? Outermost layer of dicotyledonous root. Is it cortex, epiblema, endodermis, pericycle? So it is epiblema because the outermost layer in the dicot root, we call it as epiblema. Instead of epidermis, we call it epiblema. So option B is the right answer here. Next question. What is the function of the Casparian strips present in the endodermal cells called? So Casparian strip, first of all, you have to understand that Casparian strip, we find it in dicot root. Dicot root. Where it is present in the endodermis, that is the radial and the tangential walls of endodermis will have this Casparian strip. Now what is its function? Allows water to pass through, allows air to pass through, prevent water from passing through, prevent air from passing through. It is allows water to pass through. That is the Casparian strips. So this Casparian strip, it is actually impermeable to water, meaning it doesn't allow water to pass through. Therefore, the answer is prevent water from passing through. That is the function of the Casparian strip. Next question, where does the initiation of lateral roots and vascular cambium take place in the dicotyledonous leaf? What is the question? Where does the initiation of lateral roots and vascular cambium take place in the dicotyledonous root? Epiblemma, cortex, endodermis, pericycle. So they take place in the endodermis. So from the endodermal region, usually the lateral roots and the vascular cambium and all begins there. Next question, what is the function of the conjunctive tissue in the dicotyledonous root? So conjunctive tissue is nothing but it is the tissue that is the cells present between xylem and phloem patches. So that we call it as the conjunctive tissue. What are conjunctive tissue? The cells or the parenchymatous cells which are present between the patches of xylem and phloem, because in dicots we have radial arrangement of vascular bundles, right? Wherein one patch of xylem, phloem, alternately it will be arranged. So there in between those two, there is some parenchyma cells which are being present, and that are together called as conjunctive tissue. Now, what is the question here? What is the function of the conjunctive tissue in the dicotyledonous root? Initiate lateral roots, form the cambium ring, separate xylem and phloem, connect xylem and phloem. So, usually they separate xylem and phloem. So, that is the function. So, next question. How many xylem and phloem patches are usually present in dicotyledonous root? Dicotyledonous root, usually they have 2 to 4. 2 to 4 xylem and phloem patches will be present. It's 2 to 4. It is not 5 to 6. 5 to 6 or polya condition is seen in monocotyledonous root. So next question, what is the function of the cambium ring in the dicotyledonous root? Is it initiate lateral roots, increase root length, increase root girth, form the pericycle? So cambium, they also can increase the girth of the root or size of the root, because these cambium, they develop into secondary xylem and phloem, therefore increasing the size of the root. So therefore, it increases the root girth. Next question. How does the number of xylem bundles in a monocot root compare that in a dicot root? So monocot roots have 2 xylem bundles. Monocot roots have more than 6 xylem bundles. Monocot roots have fewer xylem bundles. Monocot roots have the same number of xylem bundles. Actually, when compared to the dicot root, monocot roots have more than 6 xylem bundles. That is, they exhibit polyarch condition. So that you have to remember. This is very important. Polyarch condition, because it is one of the characteristic feature to distinguish between a dicot and a monocot. Dicots, usually they'll have only 2 to 4 xylem, but monocots will have more than 6 xylems. That is why. Next question. What is the term used to describe monocot roots that have more than 6 xylem bundles? So it is called polyarch. Next question. What is the main difference between the pith in a monocot root and a dicot root? We are talking about the pith. The pith is large and well developed in monocot root. The pith is small and poorly developed in monocot root. The pith is absent in monocot roots. The pith is located outside the vascular bundles in monocot root. It is, the pith is large and well developed in the case of monocot root when we compare in the monocot root when we compare it to the dicot root. So next question, what kind of cells are found in the hypodermis of a dicotyledonous stem? Now where is the hypodermis present? Below the epidermis. Now what kind of cells is it made up of? Which one? Dicotyledonous stem. So what kind of cells are found in the hypodermis of a dicotyledonous root? Now this hypodermis that is there, it is present where? Below the epidermis. What type of cell it is made up of? I am talking about a dicotyledonous stem. It is made up of collenchyma. Sclerenchyma, it is a monocot stem. Monocot stem. For this, the answer is collenchyma. Next question. What cells are found in the medullary rays of a dicotyledonous stem? Now, medullary rays. So, what happens here is, between the vascular bundles, there are some cells which are present, and those cells which separate one vascular bundle with that of the other, that we call it as the medullary rays. Now, what cells are found in the medullary rays of dicotyledonous stem? So, what type of cells are found in the medullary rays of dicotyledonous stem? It is parenchymatous cells which are thin-walled. Usually, they have a thin-walled parenchymatous cells. Next, which layer of the cortex is rich in starch grains? It is endodermis. So, that is why endodermis is also called as what? Starch sheath. Based on this also MCQ can be formed. So, it is called starch sheath. Next question. What surrounds each vascular bundle in the monocot stem? Parenchyma cells, sclerenchyma cells, phloem cells, xylem. So, vascular bundle means it is made up of xylem and phloem. What surrounds each vascular bundle in the monocot stem? So, it is sclerenchyma cells. So, usually around each vascular bundle that is there, we know that in monocot stem the vascular bundles are scattered, wherein the larger vascular bundles are towards the center and the smaller ones are towards the periphery. Now, surrounding each of these vascular bundles, sclerenchyma cells will be present. So, next question. What can be found within the vascular bundles of monocot stem? Water containing cavities, collenchyma cells, phloem fibers, xylem, parenchyma. So, they have water containing cavities. Next question. Which type of parenchyma possesses chloroplast and carries out photosynthesis in a dorsiventral leaf? So, dorsiventral leaf means it is a dicot leaf. Isobilateral leaf means it is a monocot leaf. It is palisade parenchyma, which is present, which are long elongated cells present just below the epidermis. It is palisade parenchyma. Next question. So, where are the stomata generally more abundant in a dorsiventral leaf? Is it vascular system, mesophyll, abaxial epidermis, adaxial epidermis? So, it is usually in the abaxial epidermis, that is lower side of the leaf, lower endodermis, wherein lots of stomata are generally distributed in the case of dorsiventral leaf, that is dicot leaf. Next question. What surrounds the vascular bundles in a dorsiventral leaf? So, in dorsiventral leaf, the vascular bundles are surrounded by bundle sheath cells. So, next question. What are the large empty colorless cells formed by certain adaxial epidermal cells in grasses called? So, what are they called? Bulliform cells. So, next question. What happens to the leaves of grasses when the bulliform cells have absorbed water and are turgid? So, when the bulliform cells have absorbed water, these cells tend to swell. So, when they tend to swell, the surface area of the leaf increases, therefore exposing the leaf surface. Meaning, option D, the leaf surface is exposed. So, that is the right answer. Now, when they become flaccid, the leaf curls inwards. That is, when they lose water, the bulliform cells, they become flaccid and the leaves curl inward, so that again too much of water is not lost for that particular purpose. So, this was about the multiple choice questions. Next, let's move on to the 3-mark question. So, one 3-mark question will appear from this particular chapter. So, first question, list the different types of tissue systems in plant body. It is epidermal tissue system, ground tissue system, vascular tissue system. So, all the 3 together will give you 3 marks. 3 tissue systems you have to mention there. Next, explain the structure of stomata. This is one of the very, very important question. Either they can ask, explain the structure of stomata, or they can ask, draw a neat label diagram of the stomatal apparatus. This is usually asked. It is very, very important. So, here you have to write the structure. So, at least 5 to 6 points is needed. The stomatal pore is closed between the 2 bean-shaped guard cells. So, enclosed, that is between the 2 bean-shaped guard cells, there is the stomatal pore that is present, and the inner walls of the guard cells will usually be thick, whereas the walls towards the outside of the guard cells are usually thin, that you have to mention. And the guard cells are surrounded by some epidermal cells, which are called as the subsidiary cells. And apart from that, you have to write, in the guard cell, they contain chloroplast and some small vacuoles will be present. And the pores of the guard cells and the subsidiary cells together, we call it as the stomatal apparatus. So much points you need to write, or 5 to 6 points if you write it is enough. So next question. This is also very, very important. Draw a neat label diagram of the stomatal apparatus. Diagram should be from the NCERT textbook, wherein you have to mark the epidermal cells. Then you need to mark the subsidiary cells, the chloroplast, the guard cells and the stomatal pore. So, totally 1, 2, 3, 4, 5 labelings are there. 5 labels should be put. Next question. Differentiate between dicot stem and monocot stem with reference to ground tissue system. Now, you have to differentiate between the two with respect to ground tissue system. Ground tissue system means with respect to the cortex, we have to differentiate. So, here the stem is solid. Here it is hollow. In monocots, it is hollow. In dicots, Below the epidermis, hypodermis is present. And what is that hypodermis made up of? Collenchyma cells. In the case of monocots, what is the hypodermis made up of? Sclerenchyma cells. Next, talking about, if you write the ground tissue system, that is enough. Or talking about the vascular bundles, you can write about the pith and all that. So, vascular bundles also have included here. So, vascular bundles are arranged in ring-like arrangement above the pith. And here the vascular bundles are scattered and they are covered by a bundle sheath, which is sclerenchymatous in nature. Next, in the dicot stem, the floral parenchyma is present, but in monocot, floreum parenchyma that is there, it is absent. In dicot, it is present. Next, the pith that is there, it is well developed and the pith is not that well developed. In dicot, vascular bundles are open and conjoined. In the case of monocots, vascular bundles are closed and conjoined. Next, in the case of dicot, layer of endodermis, it is rich in starch and therefore that layer is called as a starch sheath. But in the case of monocots, the endodermis doesn't have a starch sheath. So, these so many points you have to write. Either 3 on each side you have to write. Next question. This is also a very, very important question. Diagrammatically represent various types of vascular bundle. So, you have to draw about the radial vascular bundle. So, this is the radial vascular bundle wherein it has patches of xylem and alternating to it is patches of phloem. Next, you have to draw about the conjoint vascular bundle. So, conjoint, the meaning is conjoint means the xylem and phloem are joined together, wherein down there is xylem that is present and top it is phloem. So, this is xylem and this is phloem. Now, this is closed vascular bundle wherein no cambium between the xylem and the phloem. In the case of open vascular bundle, there is cambium between the xylem and the phloem, and open vascular bundles are usually seen in dicots and closed vascular bundles are usually seen in monocots. So much you have to draw and label. Next question. What are the important anatomical features of dicot root? So, what is there? Right from the topmost layer, we have to write about the epidermis, and then we have to write about the multilayered cortex. What is that multilayered cortex made up of? It is made up of parenchyma cells with intercellular spaces, and we have to write about the endodermis, and the endodermis is made up of what type of cells? Tightly packed barrel shaped cells, and below the endodermis what is present? Pericycle is present, and below the pericycle it has a central pith which is not that distinct, and it has 2 to 4 xylem and phloem patches, and these xylem and phloem patches are separated by layer of parenchymatous cells, and that is known as conjunctive tissue. So much you have to write. So, next question. What are the important anatomical features of monocot root? So, pith is conspicuous and large in the case of monocot root, and the number of xylem in the case of monocot root is polyarch condition, that is more than 6, and secondary growth is not seen in monocot plants because they don't have a cambium ring. So, that is why. Next question. What are the important anatomical features of dicot stem? So, the epidermis. Again here, starting from the epidermis, we have to write about the epidermis, and above the epidermis, we have to write there is a layer of cuticle that is present, and then we have to write about the endodermis, the pericycle. Then we have to write that is the cortex region is made up of hypodermal cells, and that hypodermis is nothing but it consists of collenchymatous cells, and we have to write about the endodermis, and all that needs to be written for the case of dicot stem. So much anatomical features you write, you will get marks. 3 or 4 points if you write also, it is enough. So, I have put again about the vascular bundle and all that I have written, that is ring-like arrangement they have, and conjoint open type is the vascular bundle that is present, and pith is made up of parenchymatous cells. All that you need to write. Any 3 to 4 points if you write it is enough. Next question. What are the important anatomical features of monocot stem? So, monocot stem, again the hypodermis is made up of what? Sclerenchyma cells. Vascular bundles, many are there, that is, but it is scattered, conjoint and closed vascular bundle, and they're surrounded by the ground tissue. And next, phloem parenchyma is absent in this particular case. 3 points you write, you will get 3 marks. Again here, what are the important anatomical features of dicot leaf? Again here you have to mention about the cuticle, the epidermis. You have to write that the stomata are present more on the lower epidermis than the upper epidermis. And also you have to write about the mesophyll. What is the mesophyll made up of? Parenchyma cells. What type of parenchyma cells? Palisade parenchyma and spongy parenchyma. And you have to write in the palisade parenchyma, chlorophyll pigments are present, which helps in photosynthesis. And you have to write about the vascular bundles. What are they surrounded by? They're surrounded by bundle sheath cells. Next, what are the important anatomical features of monocot leaf? Here both stomata is present on the abaxal and adaxial epidermis, which are equally distributed. And there is, they have parenchyma cells, but they do not have parenchyma cells in the form of palisade and spongy parenchyma. Bulliform cells are present, that you have to write. And how does the bulliform cell look? They are large, colorless, all that balloon shaped, all that you can write. And what is their function? To prevent excessive loss of water during summers. That is their function. So much if you write, you will get 3 marks. So much if you write, you will get 3 marks. So this was about the 3-mark questions. Apart from that, they can ask, draw a neat label diagram of dicot root, dicot stem, monocot root, monocot stem for 3 marks. That also can be asked along with these questions. So this was about the session wherein we discussed about the MCQs and the important 3-mark questions under this. So I hope you found this session useful. So we shall meet again in the coming session discussing a new chapter, doing the revision class for a new chapter. So see you in that revision session. Thank you.